So, hello everyone. I'm very glad to be able to present here our data on microglia in major depressive disorder. I do this on behalf of our team at the Netherlands Institute for Neuroscience in Amsterdam, and the paper actually is in press now in Biological Psychiatry and will appear next month. We just got a very nice introduction, I think, into microglia biology by Bart Echen, and I already announced that we will introduce a new state of microglia, just add to the many different forms of microglia we already know and hope to discuss the next days here in this meeting. But first, a short introduction about major depressive disorder, which is a severe psychiatric condition and the leading cause of disability, as we I guess all know. About a third of all patients are actually resistant to the current treatment opportunities we have. And quite often in the past, MDD has been linked to inflammation, and that is based actually on changes seen in pro and anti-inflammatory cytokines, but also on association of the disease with chronic inflammatory disorders in the patients. And we know that peripheral and central inflammation, of course, alters microglia activity, which may affect neuronal functioning through, for example, synapse stripping. And so we decided, and recent studies indeed provide already evidence that the immune status of microglia and MDD indeed is altered. But these studies were based on flow cytometry on CyTOF and not really look at the entire transcriptome of microglia in people with MDD. And that was the purpose of this work. Just a brief introduction of the psychiatric program of the Netherlands Brain Bank. Do I have here a pointer? Yeah. Here to the left, you see the abductions that have been performed over the last 4 decades. You can see that these are roughly 5,000, and most of the donors had neurological disorders or were basically controls. And 10 years ago, a psychiatric program was started that so far delivered more than 200 brains of people with psychiatric disorders. And here you can see the different disorders that have been collected, among which major depressive disorder brains, which we analyzed in our study. And here to the right, you see the current registrations of living donors, again, roughly 5,000. And you see that the share of the psychiatric disorders has grown a lot. Now we get about 30% of all donors have psychiatric disorders. And so these brain bank will become an even more important resource for studying psychiatric disorders by using tissue or cells that you immediately isolate post-mortem. For our study, we use this cohort with controls and MDD patients. You can here see that we matched them as good as possible for sex and age, and that the post-mortem delay is really very short, with 7 to 8 hours on average. We collected material from corpus callosum and occipital cortex, then processed the material by mechanical and enzymatic dissociation. Then a Pocock gradient, and this way we were able to isolate mononuclear cells, which we sorted with beads for microglia, for RNA sequencing and flow cytometry. And here you can see the immediate control that we do by flow cytometry of all the isolated cells. CD45 is a very good marker for microglia activation, and what you already see here is in the MDD cases that CD45 was actually a bit lower down in white matter as well as in gray matter, so not really showing any signs of activation so far. So then we performed bulk RNA sequencing of the isolated microglia, and you can here see that the first that the main share of the variance actually was caused by differences between white and gray matter. This is a bit as expected. On the second component here of these 10% variance, there was quite some influence of sex, female versus male, and these were genes related to either X or Y chromosomes that we neglected in the further analysis. When we then looked into differentially expressed genes, it was probably a bit of a surprise that we found no changes in white matter, and that all changes that we found actually were seen in the gray matter. And of the gray matter differentially expressed genes, 81 were downregulated and only 11 were upregulated. And you see it here also on the bottom, and they are highlighted a couple of genes that all basically are involved in immune cell functions and in phagocytosis, and that I show here in a bit more detail in this table. These are really molecules very well known, I think, to macrophage and microglia biologists. CD163 is a scavenger receptor and was a top-down regulated gene in our comparison, but also Ki67, a proliferation marker, was down, and then osteopontin, Bart mentioned it a couple of times, it is a typical molecule for microglia activation, was down in our data set, as were several FC receptors, CD14, which is involved in pattern recognition. And we also saw several related complement cascade genes to be downregulated. So when we submitted the work, the reviewers wanted us to confirm at least one of these genes by means of immunohistochemistry, for example, and we choose CD163, the scavenger receptor, which also was quite interesting because this molecule is better known for border-associated macrophages in the brain. So expression levels in microglia are lower as compared to the macrophages. But here to the right, you see nicely here macrophages at the borders of the vasculature, highly expressing CD163. But also here, a microglia very clearly with a typical cell shape is expressing the molecule. And importantly, semi-quantitation allowed us to confirm that CD163 protein expression is downregulated significantly in the MDD patients. And probably also good to notice here the diamond symbols. These are patients that committed suicide, of which we are very sure that at the moment that we basically got the brains, they were in a state of active depression. And they were just the cases with the lowest expression of CD163. We of course also did some further analysis here, IPA analysis, in which we looked for pathways that are regulated and in line with the genes that I just showed you. You see several macrophage effector functions like the effector molecule production by macrophages, but also phagocytic processes. Here probably the interplay with T cells, C127, and cytokine release by macrophages. So all these functions actually were affected in the MDD microglia. And here a WGCNA analysis showing modules that relate either or correlate either with gender or with age or with cohort. And of course, we were most interested in the ones that relate with the cohorts or with the difference between controls and MDD. And the most significant one is here this M2 group that you see here to the right with the molecular processes. And again, you see a couple of processes that all relate to phagocytosis and macrophage function. The changes we found were seen in gray matter. And in gray matter, the bodies of neurons are in close proximity to microglia. And so we wondered whether there could be an interplay between neurons and microglia involved in the processes that we see. And to test this, we focused on molecules of which we know that there are ligands on neurons that actually regulate the activity of microglia. They inhibit microglia activation states. And probably the two best known molecules in this respect are CD200 and its receptor, which has been described by John Sedgwick already in 2000. And then CD47, which is acting on CYP-alpha, thereby inhibiting microglia, work by Beth Stevens more recently. And we wondered whether these molecules, probably in the interaction of neurons and microglia, might be regulated in patients with MDD. And to test this, we started again collecting material from occipital cortex. Here you see that the cell material that we collected. And we processed the tissue for either RT-PCR or did ultracentrifugation and separating myelin from the synaptosomes that we tested by Western blot. To start with the RT-qPCR, you see here the regulation of gene expression of the ligand genes in the neurons, CD47, which was up, and CD200, which was also upregulated in the neurons, and for CD200, the difference was significant. Then the Western blot, where we first looked into a molecule related with synapses that we really found enriched in our ultracentrifuged preparation, and that was absent in the myelin fraction. So we really look here at synapse proteins, and we tested by Western blotting the presence of CD47 and CD200. And you can see that the molecules are here upregulated in MDD, at least a trend is also seen for CD200. And yeah, here you can see the Western blot of both molecules. So it really seems that we see that the ligands that inhibit microglia activity are upregulated in neurons in the gray matter, which may explain the changes that we see in microglia biology underneath. So conclusions. MDD-associated microglia downregulate immune and phagocytosis-related genes. The results are in line with some former findings done by CyTOF, by flow cytometry, by RT-qPCR, performed by the group of Lotte Witte, who basically also showed a more dampened state of microglia. And importantly, in their work, they also show some upregulation of homeostatic genes like TMR119 and the factor kinase receptor 1. The exclusive presence of the differences that we see in gray matter microglia and not in white matter microglia suggests that the changes may be induced by neuronal interference and by molecules that I just, or receptor ligand pairs that I just showed you, like CD47 and CD200. And we conclude that MDD actually extends the wide scale of microglia signatures with an apathic or suppressed state. You can a bit argue about what is the best wording for describing this state. And our paper will be accompanied by a commentary by Yass Yemia, a psychiatrist from Israel, and he uses the term suppressed depression disorder associated microglia, basically for the phenotypes that we describe in our paper, and that fits with the data of Lotte Witte. And he also shows the difference of this state of microglia as compared to an activated depression disorder associated microglia state that has been reported in some other studies. So this is certainly something we can discuss here during this meeting, how these new states fit into the wider horizon of microglia developmental and disease-associated states. And this is my last slide here showing the main actors on this study, Karel Streepstra, Mark Mizet, and the head of the research team at the Netherlands Institute for Neuroscience, Inge Houtinger. And I think with this, I finish my talk, and thank you for your attention.
Thanks very much. Questions? Starting.
I had a question.
the data that you showed, CD200 upregulated and CD47 upregulated, can you comment on what happens to the receptors?
In our transcriptomic data, we do not see changes in the expression of the receptors on the microglia. So it seems here that really, in particular, the ligands on the neurons are upregulated.
Okay, thank you.
Thank you very much for your talk. I actually had two questions. So one, you hinted at that there is a bit of a difference between sexes, so males and females. I was wondering if you could just comment a little bit more on that. And I think in your final slide as well, you showed that there is a higher numbers of sort of those suppressed depression microglia, as you call them, in females versus males. So if you could just comment on those gender differences.
Yeah, the sex differences that we found were basically based on X and Y chromosome located genes, and these genes we excluded from the father analysis just to basically be sure that we look at cohort differences. Yeah.
Fair enough. And my other question was about the medication state of the patients, given some of the psychiatric drugs seem to affect the immune system to some extent as well. So I was wondering what the medication status of the patients was.
No, I'm not a psychiatrist myself. I mean, this is a question that we got very often with these materials that we obtain here from the Netherlands Brain Bank. I mean, the patients for sure took a bunch of medication throughout their life. I'm not sure to what extent the medication may affect here gene expression. That we can't exclude that.
Thank you.
Hi. In one of your early slides, you said that CD45 was a marker of microglial activation. Yeah. CD45 is a receptor tyrosine phosphatase. Do you know anything about tyrosine phosphorylation-dependent signaling in those cells?
No, not really. I think we really considered here basically as a marker for the activation state of microglia, not really looking into the molecular biology behind this molecule. But it's a very robust marker that you also see upregulated, for example, in microglia from MS patients.
Yeah, I noticed one of your differentially expressed genes between normal and the condition, major depressive disorder, was CD163, wasn't it?
Yeah.
Right? And when you showed the staining, it seemed to be associated, particularly as you pointed out, with the blood vessels. Mm-hmm. And I just wondered, is there any distinction in any of these transcriptomic studies between microglia and perivascular macrophages? Because are we looking at perivascular macrophages in this situation? Could those be the interesting target population?
Perivascular macrophages, I mean, this is bulk RNA sequence. No, I think we can exclude it. I mean, these are sorted microglia, and we carefully looked in our transcriptomic data set for different other cell types or for markers of other cell types to be sure on the purity of the cells. And I think we are pretty sure that we are really looking here at my hand. Here you can see it's the microglia genes that we see in our data set, and But for example, here vasculature macrophages, but here we also tested a couple of true macrophage markers that are expressed by brain macrophages and that are not really present in the data set.
The perivascular macrophages is a very distinct population, though, aren't they? I mean, where do they sit on the? Are they microglia? Are they macrophages? Are they sort of a subpopulation of microglia? Maybe some other people know the answer to that question. I know you express CD206. I mean,
yeah.
The connection with the blood vessels is interesting, though. I just wondered if the blood vessels also driving some of this disease process.
But I mean, essentially, we can't exclude that CD163 is also regulated on these perivascular macrophages. But in our transcriptomic data set, we should not see patterns that are derived by macrophages. I mean, based on cell purity.
I was just wondering if you could comment on which areas of the brain you looked at and whether your findings are across the brain or in specific areas, because something like depression is quite difficult to pin down to particular brain regions?
I mean, we choose these regions actually based on guidance that we got from psychiatrists to look here. I know that Lotte de Witte also compared more different brain regions with her approaches, and I think she found some molecular changes reported by her across regions. But that of course is a very relevant and also difficult question to choose the proper brain regions in these postmortem research.
Great, thanks very much. We've got to move on.